Welcome to the fascinating world of microbiology, where we explore the lives of organisms too small to be seen with the naked eye. These microscopic wonders, called microbes, are everywhere. In the air we breathe, the food we eat, and even within our own bodies. Microbiology is the study of these tiny organisms, including bacteria, viruses, fungi, protozoa, and algae. Although invisible without assistance, these minute life forms play a crucial role in maintaining life on Earth. Microorganisms are the foundation of all ecosystems. They drive nutrient cycles, decompose organic matter, and form the basis of food chains. But microbes are not just silent partners in sustaining life. They also have a profound impact on human health, both positively and negatively. From the development of antibiotics to the use of microbes in biotechnology, microbiology has led to significant advancements in medicine, agriculture, and environmental science. Join us as we uncover the secrets of the unseen world that surrounds us. The journey into the microscopic world began with curious minds and rudimentary tools. One such pioneer was Antony van Leeuwenhoek, a Dutch draper with an insatiable thirst for knowledge. In the late 17th century, Leeuwenhoek crafted simple single-lens microscopes that achieved remarkable magnification. With these instruments, he became the first person to observe and describe bacteria and protozoa, which he called animalcules. Another key figure in early microbiology was Robert Hooke, an English scientist who coined the term cell in 1665. These early pioneers, limited by the technology of their time, could only marvel at the existence of this unseen world. The invention and refinement of the microscope were instrumental in advancing the field of microbiology. Microscopes act as windows into the invisible world, allowing us to observe the intricate structures and behaviors of microorganisms. There are several types of microscopes, each with its own unique capabilities. The light microscope, a direct descendant of Leeuwenhoek's and Hooke's instruments, uses visible light and a series of lenses to magnify specimens. For visualizing even smaller structures, such as viruses and internal cellular components, scientists rely on electron microscopes. Electron microscopes use beams of electrons, which have a much shorter wavelength, to achieve significantly higher magnification and resolution. More recently, advances in microscopy have led to the development of fluorescence microscopy, confocal microscopy, and atomic force microscopy. The ongoing development of new microscopy tools continues to expand our ability to explore the microscopic world in ever greater detail. For centuries, the prevailing belief was that living organisms could arise spontaneously from non-living matter. This idea, known as spontaneous generation, was based on observations such as the appearance of maggots on rotting meat or the growth of mold on bread. However, the development of the microscope and the careful experiments of scientists like Francesco Redi and Louis Pasteur challenged this long-held notion. In the 17th century, Redi demonstrated that maggots did not arise spontaneously from meat, but were actually the larvae of flies. He covered jars containing meat with gauze, preventing flies from laying eggs, and observed that no maggots appeared. It was Louis Pasteur, a French chemist and microbiologist, who dealt the final blow to spontaneous generation in the 19th century. Pasteur's work not only refuted spontaneous generation, but also established the principle of biogenesis, which states that living organisms can only arise from pre-existing living organisms. Section 5. The germ theory of disease. Before the acceptance of the germ theory, diseases were often attributed to supernatural forces, imbalances in bodily humors or miasmas, which were poisonous vapors thought to be released from decaying organic matter. However, the work of Louis Pasteur, Robert Koch, and other pioneers of microbiology led to the revolutionary idea that microorganisms could cause disease. Pasteur's research on fermentation and silkworm diseases demonstrated that specific microorganisms were responsible for these ailments. He proposed that germs could also cause diseases in humans, a concept that initially met with resistance from the medical community. Robert Koch, a German physician, made significant contributions to the germ theory by developing techniques to isolate and identify specific pathogens. Koch's postulates, a set of criteria for establishing a causative link between a microbe and a disease, became the cornerstone of medical microbiology. Section 6. Koch's postulates, pillars of medical microbiology. Koch's postulates, formulated in the late 19th century, provided a systematic framework for determining the causative agent of a disease. These 4 criteria are essential for establishing a causal relationship between a suspected pathogen and a specific disease. First, the suspected pathogen must be present in all cases of the disease and absent from healthy individuals. Second, the suspected pathogen must be isolated from the diseased host and grown in pure culture. Third, the cultured pathogen must cause the same disease when inoculated into a healthy, susceptible host. Fourth, the same pathogen must be re-isolated from the experimentally infected host. Koch's postulates revolutionized the study of infectious diseases and laid the foundation for modern medical microbiology. They provided a rigorous scientific method for identifying the causative agents of diseases, leading to the development of effective treatments and preventive measures. Section 7. The vast and diverse world of microbes. The microbial world is incredibly diverse, encompassing a vast array of organisms with different sizes, shapes, metabolisms, and lifestyles. Microorganisms can be found in virtually every environment on Earth, from the depths of the oceans to the highest mountains. They have evolved a remarkable range of adaptations to thrive in extreme conditions, such as hot springs, acidic environments, and highly saline waters. Microorganisms are broadly classified into 5 major groups: bacteria, viruses, fungi, protozoa, and algae. Bacteria are prokaryotic organisms, meaning they lack a true nucleus and other membrane-bound organelles. They are incredibly abundant and play crucial roles in nutrient cycling, decomposition, and human health. Viruses, on the other hand, are not considered true living organisms because they cannot reproduce independently. They are essentially packets of genetic material enclosed in a protein coat and require a host cell to replicate. Fungi are eukaryotic organisms that obtain nutrients by absorbing organic matter from their surroundings. They include yeasts, molds, and mushrooms, and play important roles in decomposition and nutrient cycling. Protozoa are unicellular eukaryotic organisms that exhibit animal-like characteristics, such as motility and predation. They are found in aquatic and terrestrial environments and play important roles in food webs. Some protozoa are parasitic, causing diseases such as malaria and amoebic dysentery. Algae are a diverse group of photosynthetic organisms that range in size from microscopic single-celled species to large multicellular seaweeds. Section 8. Microbes, friends or foes. Microbes have a complex relationship with humans, acting as both beneficial partners and potential pathogens. The vast majority of microbes are harmless or even beneficial to human health. Our bodies are home to trillions of microbes, collectively known as the human microbiota, which play crucial roles in digestion, immune system development, and protection against pathogens. Beneficial microbes are also used in various industries, including food production, biotechnology, and environmental remediation. For example, bacteria are used to produce yogurt, cheese, and other fermented foods, while yeasts are essential for bread making and alcoholic beverages. Microbes are also used to produce antibiotics, enzymes, vitamins, and other valuable products. However, some microbes are pathogenic and can cause a wide range of diseases in humans, animals, and plants. These diseases can range in severity from mild infections to life-threatening illnesses. Understanding the mechanisms of microbial pathogenesis is crucial for developing effective treatments and preventive measures. Section 9. Conclusion. The unseen world that shapes our lives. The world of microbiology, though invisible to the naked eye, is teeming with life and has a profound impact on our planet and ourselves. From the pioneers who first glimpsed this hidden realm to the cutting-edge technologies that continue to unveil its secrets, our understanding of microbes has come a long way. We have learned that microbes are not merely agents of disease, but also essential partners in maintaining life on Earth. They drive nutrient cycles, decompose organic matter, and form the basis of food chains. As we continue to explore the microbial world, we are constantly making new discoveries that challenge our understanding of life itself. From the identification of novel pathogens to the development of innovative antimicrobial therapies, microbiology continues to shape our world in profound ways.